Friends, myself, Dr. P. Samuel, Assistant Professor, Department of Biotechnology of Ayanadar Janakiammal College, Sivagasi. Through this video tutorial, I am going to share something about what are stem cells, right? So, this is the topic I wish to convey to everyone, an interesting topic. So, listen to me very carefully. So, let us begin this lecture. As we all know that all organisms, right, starting from human, animals, wild animals, even small insects, even submicroscopic organisms are made up of cells, right? So, this is a very general thing, we know this. Every organism is made up of cell, starting from human being, wild animal, insects, even lower microscopic organisms are made up of cells. Fine. And how can we define a cell? A cell can be defined as fundamental unit of a biological system. So, this is what the definition which we know from our lower classes. So, a cell can be defined as fundamental unit of a biological system. So, as we go in an ordered pattern, every organism, right, as we discussed in the last slide, starting from a most primitive organism like bacteria to well developed organism like human being, their ultimate structure is called cells, right? Cells are grouped together to form tissue, tissue are grouped together to form organs, organs they are grouped to form organ system, and finally the human body. So ultimately the cells are bricks, I mean the fundamental units, right? Fundamental units. So whatever the damage caused to the cell will definitely reflect in the complete biological system. So it is very essential to keep the cells energetic and to through all means it is our responsibility to keep the cells healthy, right? So in a similar way, human body consists of nearly 200 different types of cells. This is a very interesting point I want to tell you. Human body consists of 200 different types of cells. In the same way, each cell is capable of doing a specific function. Every cell has specific function. In the same way, the whole human body contains 30 trillion cells. 30 trillion cells. Imagine how complex the biological system is, right? For your information, I wish to tell that the cells present in the heart muscle, right? The principal type of cell present in the heart muscle is called cardiomyocytes, right? Cardiomyocyte, which contain more number of mitochondria. Why? Because heart cells are continuously pumping cells. It requires more energy. So for that reason, heart cells are containing rich number of mitochondria, right? Well, see, these are all the very common types of cells. We know that RBC, WBC, and platelets. So, what is the role of RBC? They carry oxygen. What is the role of WBC? They fight with antigen. What is the role of platelets? They are responsible for blood clotting. Right? Each cell has its own function. Right? Each cell has its own function, which means I will tell you in detail. Oxygen will not be carried by a WBC cell. oxygen will not be carried by WBC. In the same way, antigens are not neutralized by platelets. So this is what I tell you, right? Each cell has its specific function, right? The second point, each cell has its specific function, and the lifespan of the cells are also limited, right? Not all the cells are living for all the years, right? So nowadays the average lifespan of a human being reaches 60, right? No cells will persist or will survive in the biological system up to 60 years. Every cell, which means all the 30 trillion cell, has a limited lifespan, right? Here I am giving two examples, right? RBC has 90 to 120 days. In the same way, WBC has 3 to 4 weeks. So what happened after completing this life cycle, lifetime period? So after this stipulated time period, the cells are taken to the graveyard. For example, in case of RBC, the damaged RBC or aged RBC are taken to spleen for further destruction or for complete destruction. So after completing its life cycle, the cell has to be destroyed. So at the periodical cycle, considerable amount of cells will be lost from the biological system. How those lost cells are replaced, or how those damaged cells are being replenished by the biological system, here the question is, right? Let's discuss it. See, for every cell type, right, after a particular time period, the cells are taken to the spleen where they are destroyed. At the same time, the body has to replace the lost cells or damaged cells at a regular interval. Otherwise, it will definitely result in a clinical manifestation. For example, if the number of RBC is decreased, the condition is called anemia. In the same way, if the number of RBC is increased, it is called polycythemia. So both increased number of cell or decreased number of cell could result in a clinical manifestation. So the cell number has to be maintained in a homeostatic stage, right? Now what I want to tell you is from where these cells are originated from. So the answer is not bone. Don't right? Bone is actually not the source. Just for your reference I am giving. So within the bone, right, you could find a reddish brown color material. The material is called bone marrow. Actually, this structure is very important. This is acting as the source for all the cells to multiply or to proliferate. So this material is present within the bones. Actually, these are called as mother cells, from which all other cells are differentiated, right? So from the bone marrow, there are two principal pathways are developed. One is called lymphoid lineage, which could result in lymphocytes, right? Lymphocytes. And myeloid lineage result in the growth of blood cells. Myeloid lineage could result in the development or growth and development of blood cells. So remember these two things. Okay. So for all the cell types present in the human body, I mean for all the 200 different types of cells, the fundamental platform, right, is the bone marrow. So from the bone marrow, all other cells will grow, right, develop, multiply, differentiate and everything, right? Okay. Now let's talk about the stem cells, the core portion of the presentation. So stem cells are known for its two unique properties. One is they are able to self renew themselves. They are able to self renew themselves. Second point, they are able to differentiate. They are able to differentiate. And these 2 points are very unique for the stem cells. And based on the 2 points, stem cells are attracted by many researchers across the globe. The two principal type of stem cells are adult stem cells, another one is called embryonic stem cell. So through this presentation, I wish to tell you about, I wish to give a special emphasis on the embryonic stem cells, right? So to begin with, what is an embryonic stem cell? You should know what is a male gamete and what is a female gamete. So this is actually a male gamete. We used to call it as a sperm cell, which is motile, and this one is called the female gamete, called as ovum, right? This is male gamete and this is female gamete, right? The process of fusion of this gamete, right? So sperm cell will move towards the ovum, right? So you could see the motility, right? So they started to move towards the female gamete. Finally, it fuses with the female gamete, right? And the process is called fertilization, fusion of gametes. Naturally, this process is taking place within the fallopian tube, but in the laboratory, the same process is taking place in a test tube, right? In a test tube. So this process will usually be carried out in assisted reproductive units. So after fertilization, during fertilization process, from the male gamete, the genetic material will be liberated or it will be inserted into the female gamete cell, and this structure is called zygote, right? The fertilized egg is called zygote. This is called as zygote. Once the embryo is formed, right, it started to undergo repeated cleavage across various planes. Repeated cleavage, 2 cell stage, 4 cell stage, 8 cell, 16, right? So like this the cleavage takes place. So exactly after 5 to 7 days of fertilization process, these embryo will contain small structures within them. They are called as embryonic stem cells. These individual cells are called as embryonic stem cells. This one, right? This one. These are called as embryonic stem cells. And these are very essential. These are mainly attracted by the people for its medical use or for its clinical use, right? Fine. And after these structures are formed inside the embryo, using a pipette, right? The structures are removed from the cell and they are transported into the nutrient medium. Nutrient medium means what? It contains essential growth substances, for example, protein and other components which are required for the growth of cells, right? This contain minerals, proteins, right, vitamins, right, everything. All these things collectively support the growth and establishment of the stem cells. So like this the cells proliferate because it is a nutrient medium. Upon utilizing the media, the cells started to grow and cover the entire surface of the medium, right? So After growth, here it is an important process, a chemical substance will be added. The chemical substances are collectively called as chemokines, which stimulate, right? Chemokines which stimulate the differentiation of stem cell, right? A functionless cell is transformed into a specialized cell. I mean, a cell with a known function, a cell with a unique function. This is called differentiation. How this differentiation is achieved? The differentiation process is achieved through the addition of chemicals, addition of chemokines into the nutrient media, right? After differentiation, the differentiated stem cells can be used to replace the damaged cells present in brain, right? It can be used to replace the damaged cells present in your liver. The same can be used to replace damaged cells in the lungs. In the same way, it can also be used to replace the cells of the kidney. So, these differentiated stem cells are medically important cells which could be able to replace the damaged cells, thereby the original function of the organ can be retained, right? After this process, this is called cell based therapy, right? In simple, people used to call it as cell based therapy. So the excess stem cells or the unused stem cells are not what's that? Is not discarded, right? It is not thrown into the crash, not like that. The excess embryonic stem cells are put it in a sucrose solution and kept in cryopreservation process, right, in a specially designed vial called cryovials, which could able to preserve the stem cells for longer period, right? Because why these stem cells are stored in the laboratory? Because of its potential medical applications, right? Another important thing I want to tell you, for research purpose, right, the usage of embryo is strictly prohibited. Why? Because embryo is equivalent to a life. So mishandling will definitely leads to death of the embryo, right? Mishandling will definitely leads to death of the embryo. So to avoid the legal action, the laboratory should get consent from the donors, right? What the main thing is, consent from the donor, which means the supplier of a sperm cell and ovum cell, right? Without the consent, right, the laboratory should not or should never continue this process, because it is a violation of Stem Cell Research Act. This will definitely bring legal consequences on the company. So no company will proceed with stem cell research without the consent obtained from the donors, right? Thank you for watching this lecture. For your queries, please mail your questions at drsamuelponpondian@gmail.com or catch me at the mobile number given in the slide. Thank you so much.